Okay. Hello dear students, today we are going to talk about on the topic of synapse. So at the end of this session you will be able to define the synapse, know what are the types of synapse, you will be able to draw a neat label diagram of synapse, know the steps in synaptic transmission and the properties of synapse. So first coming to the definition of synapse. So synapse is a junction between the two neurons. So two different, we know the parts of the neuron, we have an axon, we have dendrite and we have the soma. So depending on what part of the axon is taking part in the synaptic connection, we can divide the synapse anatomically into different parts. So first by definition, as I told you, synapse is the junction between the two neurons. Sherrington was the first person to give this term synapse between the two neurons. Now coming to the anatomical types of the synapse, depending upon what part of the neuron takes part in the synapse, we have the axonic synapse, where the axon of one neuron synapses with the axon of another neuron. Then we have exodendritic synapse, which is the most common type of synapse, where the axon of one neuron makes synaptic connections with the dendrite. Then we have exosomatic synapse, where the axon of one neuron makes synaptic connections with the soma of another neuron. And lastly, we have the dendrodendritic synapse, which is the most rare one, where the dendrites of one neuron make synaptic connection with the another dendrite of another neuron. Now that is the anatomical classification of the synapse. Now what is the need of the synapse? Synapse is very important as it transmits impulse from one neuron to the another neuron, and this transmission of impulse is required for processing whatever information is coming to the brain. So for transmission of the information, for processing of the information, especially when we talk about learning and memory in different areas of the brain, synapse is very important. Now we have done the anatomical classification. Now we can also divide or have different types of physiological classification for the synapse. So we can have electrical synapse, depending upon how the impulse is transmitted across the synapse. So where the impulse is transmitted using an electrical transmission, that is called as an electrical synapse, which we see for example in the gap junctions. Then we have chemical type of synapse, where a chemical neurotransmitter is used to transmit the information from one neuron to the another neuron, or you can have conjoint, where both electrical and the chemical transmission is used across the synapse. So now we are going to talk about the structure of the synapse. Now the synapse involves two different neurons. So we will have one neuron which is the presynaptic neuron, and we have another neuron which is called as the postsynaptic neuron. Now what is a presynaptic neuron? This is the neuron which is going to conduct the action potential, and impulse is generated through this neuron to the another neuron which is your postsynaptic neuron. Now this presynaptic neuron, the end of it's an exodendritic synapse. So the end of this axon forms a bulbous enlargement. This is called as a terminal button. Now within this terminal button you have different types of vesicles. These vesicles store the neurotransmitter. Since this is going to be a chemical type of synapse, so we have vesicles within the presynaptic terminal which contains neurotransmitters. Now the size and shape of these vesicles will be different depending upon the type of neurotransmitter which is stored within them. So it could be small, large, it could be sparsely dense, or it could be greatly dense, depending again on the transmitters. So various neurotransmitters can be acetylcholine, it can be a glutamate, it can be GABA, glycine. So there could be n variety of neurotransmitters which are present within the presynaptic bulb. Now apart from the vesicles, so you have another structure which you need to draw is the mitochondria, which supplies ATP, because these vesicles are going to release the neurotransmitter into this part between the two neurons, which is called as a synaptic cleft. So we require ATP, which is provided by the mitochondria to release the neurotransmitter from the vesicles into the cleft. Another important point here is, in the diagram you need to remember, is the voltage gated calcium channels. So calcium is a very important component again for release of the neurotransmitter into the synaptic cleft. So when the voltage gated calcium channels open, there will be calcium influx, and that will cause the vesicles to release the transmitter into the synaptic cleft via exocytosis. Then another component which is present in the presynaptic terminal are the microtubules. These microtubules help in transporting the vesicles which are filled with neurotransmitter into the synaptic trough. So that is about the presynaptic neuron. Then coming to the next part of the synapse is the synaptic cleft. Now this synaptic cleft is around 20 to 30 nanometers in depth, that is how much is the space between the two neurons, and this synaptic cleft is where the neurotransmitter is released by the vesicles. Now once the neurotransmitter is released by the vesicles, this binds or brings about potential changes in the postsynaptic neuron. Now this postsynaptic neuron, on its membrane, it has ion channels. So there could be different type of ion channels, and the receptor will bind to a specific ion channel and bring about change in the postsynaptic neuron. The vesicles which release a neurotransmitter can be taken back and recycled via the various mechanisms which we will study later, and can reuse the neurotransmitter, or sometimes the neurotransmitter can diffuse into the extracellular fluid. So this is the structure of the synapse, which includes the presynaptic neuron, the synaptic cleft and the postsynaptic neuron. So we have seen the structure of the synapse. Now we will talk about the synaptic transmission. Now the first step in synaptic transmission is arrival of the action potential in the presynaptic terminal. Now once the action potential arrives in the presynaptic terminal, what it will cause? It will cause depolarization of the synaptic membrane in the presynaptic neuron. Now depolarization of this membrane causes opening of voltage gated calcium channels. Now once the voltage gated calcium channels open, they cause influx of calcium inside the presynaptic terminal. Now that is just step 3. Now once calcium influx occurs, it causes fusion of these vesicles on the presynaptic membrane. Now this presynaptic membrane contains the active zones. Now these zones are areas where the synaptic vesicle will bind to this membrane, and from this active sites, the neurotransmitter will be released into the synaptic cleft. So once the action potential arrives, causing depolarization of the membrane, the depolarization in turn opens up voltage gated calcium channels. Calcium moves inside the presynaptic terminal, and because of that, calcium causes fusion of the vesicles on the active zone. This is the active zone. Now the active zone contains also extra proteins which will cause the vesicles to undergo contraction and release the neurotransmitter via exocytosis into the synaptic cleft. Now once these neurotransmitters are released in the cleft, they bind to the receptors on the postsynaptic neuron, and that causes opening up of ion channels. So there could be either influx of sodium or influx of calcium, or if they open up chloride channels or potassium channels, chloride will move inward or potassium will move outward. So depending upon what type of ion channels are present, it will create synaptic potentials. Okay, so now we will be drawing a flow chart on synaptic transmission. So in the exam, whenever you have a question, you should be able to draw a neat label diagram, and the diagram should be accompanied by a flow chart which will help you to remember these things. So first is arrival of action potential at the presynaptic terminal. Okay. Now once action potential arrives at the presynaptic terminal, it causes depolarization of the terminal button. Now once the terminal button is depolarized, this causes opening of voltage gated calcium channels open, causing calcium influx. If the calcium influx occurs, then the calcium causes the vesicles to fuse with the membrane and release the neurotransmitter by exocytosis. Okay, so there is a release of neurotransmitter by exocytosis. Then the fifth point, the neurotransmitters will bind to receptors on the postsynaptic membrane, and lastly, opening of ion channels and generation of synaptic potential. And that will generate a synaptic potential. So those are the steps: arrival of the action potential at the presynaptic terminal, depolarization of the presynaptic membrane, opening of voltage gated calcium channels causing calcium influx. The calcium in turn causes fusion of vesicles in the presynaptic membrane and release of neurotransmitter in the synaptic cleft. The neurotransmitter binds to the receptors on the postsynaptic membrane, opening of ion channels on the postsynaptic membrane, and development of synaptic potential. Now after seeing the steps of synaptic transmission, now we will talk about the synaptic potentials. Now a single action potential arriving in the presynaptic neuron cannot propagate an action potential in the postsynaptic neuron. So the end result, what is produced when the neurotransmitter is released, are the synaptic potentials. So this synaptic potentials which can be excitatory, which is called as the excitatory postsynaptic potential. It is a transient potential which is produced in the postsynaptic membrane. It is usually a depolarizing potential, or it can produce a hyperpolarizing response in the postsynaptic neuron, which is called as a inhibitory postsynaptic potential. So the two potentials we have, the EPSP and IPSP. Now we will study what is the mechanism of production of EPSP and IPSP, or how the electrical events are produced in the postsynaptic membrane. Now, so first potential, as I told you, action potential generated a single stimulus coming into the presynaptic neuron cannot initiate a propagated action potential in the postsynaptic neuron. In turn, it produces synaptic potentials. So it can either produce an EPSP or an IPSP. Now when the neurotransmitter binds to either to the receptors in the postsynaptic membrane, it will open up either sodium channels or calcium channels. These are chemical mediated channels. Sodium and calcium channels open up, and when these channels open up, as they are positive ions, they cause depolarization of the postsynaptic membrane. So that produces a transient depolarization potential, and when this membrane is depolarized, the postsynaptic neuron, at this point it is excitable to the next stimulus which it is coming. Hence it is called as an excitatory postsynaptic potential. So this is the EPSP which is produced because of opening of sodium and calcium channels, and it causes depolarization of the postsynaptic membrane. Now also opening or closure of the potassium channels can also bring about similar transient depolarization of the postsynaptic membrane, because when the potassium channels close up, potassium remains, it is a positive ion, it remains inside the postsynaptic membrane and causing a transient depolarization of the postsynaptic membrane. So that is an EPSP. At this point the neuron is highly excitable to the next incoming stimulus. Now just opposite to that, we have the inhibitory postsynaptic potential. Now the same again, when the impulse arrives at the postsynaptic terminal, at the presynaptic terminal, it opens up certain ion channels. Now when the neurotransmitter opens up ion channels like chloride or potassium, it causes hyperpolarization of the postsynaptic membrane. Now the chloride ions channel open up, chloride will move inside. So negative ion coming inside will cause a hyperpolarizing current. Similarly, when potassium channels open up, potassium diffuses outside the cell, again creating a hyperpolarizing current in the postsynaptic membrane, and that is your IPSP. Now at this point the neuron, it is in a refractory period, it will not respond to the stimulus which is coming towards from the presynaptic neuron. Hence it is called as a inhibitory postsynaptic potential. Also closure of sodium and calcium channels can cause a similar hyperpolarizing response in the postsynaptic membrane. So we have the EPSP and the IPSP. These are graded potentials which are produced in the postsynaptic membrane when a single stimulus arises from the presynaptic membrane. Now the synaptic potentials have been produced, whether it is EPSP or IPSP. We will talk about how this causes propagation of action potential in the postsynaptic neuron. Now a single neuron is going to receive, this postsynaptic neuron receives electrical stimulus from many presynaptic neurons. So there will be IPSP will be produced, which is red in color, and you have the EPSP or green in color. So many EPSPs will be produced on the postsynaptic neuron, as well as IPSPs will be produced. Now what is the soma doing? Because the soma is receiving this all IPSP and EPSP, it is going to summate. So what is the role of the soma is to summate all the electrical potentials which have been generated, whether it is EPSP and IPSP, and finally the algebraic sum of these two potentials will decide whether a propagated action potential will occur in the postsynaptic membrane. So once if reaches the threshold level, then it is going to stimulate the axon hillock and the initial segment and cause depolarization here. So once this reaches the threshold level, it causes depolarization of the axon hillock and the initial segment, and this in turn will cause propagated action potential in the postsynaptic neuron. So that is how the impulse is generated in the postsynaptic neuron. Okay, so now we will just take an example of how this summation is going to occur on the soma. Suppose the EPSP produced on the postsynaptic neuron has a voltage of around 16 millivolts and the IPSP produced has a voltage of minus 30 millivolts. So the algebraic sum total, we will have a 30 millivolt threshold will be created. So this 30 millivolt is sufficient to stimulate the axon hillock and the initial segment, and that will cause propagation of action potential in the postsynaptic neuron. Now we will start with the properties of synapse. There are in total 10 properties which we are going to study. So first, coming to the property of one-way conduction, which is also called as a law of forward conduction. Now in this property, we know that the conduction of impulse will always be from the presynaptic axon to the postsynaptic neuron. So this one-way conduction is important because it will allow orderly conduction of the impulses from presynaptic neuron to the postsynaptic neuron. Now when the impulse is conducted in this direction, that is in retrograde direction, some amount of impulse, if it goes in the antigrade direction, that it goes upwards towards the soma, in the soma this impulse will die out. So the impulse will always be conducted from the axon terminal of the presynaptic neuron to the postsynaptic neuron, because in the presynaptic neuron only you have vesicles here. So at the synaptic junction in the presynaptic neuron there are vesicles which are going to release a neurotransmitter and conduct the impulse in the postsynaptic neuron. The membrane of the postsynaptic neuron does not have any neurotransmitter to conduct. So impulse will be always conducted in this direction, and that is important for the orderly conduction of impulse and for the proper processing of the neuronal information. So the next property now is the synaptic delay. Synaptic delay is around 0.5 milliseconds. Now this delay occurs because of transmission of impulse from the presynaptic to the postsynaptic neuron. So the events like the releasing of the neurotransmitter from the presynaptic terminal to the postsynaptic terminal is responsible for this delay of 0.5 milliseconds. Now what is the significance of the synaptic delay? This allows you to determine how many number of synapses are taking part in a reflex. So whether the reflex is a monosynaptic reflex or it is a polysynaptic reflex can be determined by the time it requires for the impulse to conduct from the presynaptic neuron to the postsynaptic neuron. So if more number of neurons are involved, the reflex delay will be much greater, and that will be a polysynaptic reflex. Our next property is convergence and divergence. What do you mean by convergence? So when one single neuron, many neurons are converging on one single neuron. That is a property of convergence. Okay, so this example can be given when the alpha motor neuron in the spinal cord, it receives afferents say from the muscle, plus this motor neuron is also receiving inputs coming from the descending tracts. So there is convergence of signal coming from the dorsal root as well as from the cortex. So this is an example of convergence which we see. Then coming to the property of divergence. Now divergence means one single neuron making synaptic connections to many neurons. So we have one neuron converging on many postsynaptic neurons. So again the example will be the sensory signals as they travel up, your sensory tracts as they travel up to the cortex, they give out signals to many other areas of the brain as well. So that is an example of divergence. Now this obviously this is going to cause amplification of the signals as it passes up to many areas. So divergence is required, so it amplifies the signals as you move up, it spreads to many areas and you can amplify their response. So the divergence causes amplification of the signal, whereas convergence causes effective stimulation of the signal, as many impulses will come and facilitate this neuron and brings about effective either stimulation or inhibition of the neuron. Okay, now the next property is summation. Now summation can be two types, temporal summation and spatial summation. Now first we will see what is spatial summation. Now in a spatial summation, one single presynaptic neuron, we have already studied, when one single presynaptic neuron stimulates a postsynaptic neuron, it will produce an EPSP because it cannot produce an action potential. So when you stimulate, say you are stimulating a neuron, it stimulates the postsynaptic neuron X. So it is the electrical gradient is not sufficient enough to stimulate X. Now when you simultaneously stimulate all these three neurons, the EPS will summate and finally an action potential will be produced in X. So this type of summation is called as a spatial summation, where many presynaptic neurons, the EPSP generated by these neurons summates together to reach a firing level and stimulates the postsynaptic neuron. Then coming to the temporal summation. Now temporal summation, again one single presynaptic neuron does not generate sufficient potential in the neuron X to cause action potential. But if you stimulate the A neuron repeatedly, what will happen? The EPSP which was generated by the first stimulus will be added up to the subsequent stimulus. But what is the condition? That the subsequent stimulus should be applied before the potential in X dies out. So when you stimulate repeatedly, neuron A will be stimulated many times, one after the another. The EPSPs produced will be added up again, and that will cause stimulation of the neuron X. So that is a temporal summation, that is subsequent stimulus, same neuron is stimulated again and again, whereas in many presynaptic neurons summate to stimulate the postsynaptic neuron. The next property is property of occlusion. Now here one presynaptic nerve fiber or one afferent fiber, say fiber A, it stimulates 3 neurons. Okay, so one single fiber A is stimulating 3 neurons X, Y and Z. When we stimulate B, it stimulates other 3 neurons. So now when both A and B are simultaneously stimulated, say I will write down, this A stimulates 3 neurons when it's and B it stimulates 3 neurons. So responses of A is of 3, even B is stimulating 3. But when we stimulate both A and B together, the response is of 5 neurons. Now you can see here there is one neuron which is overlapping. So because of this overlap, there is a common neuron which is innervated by both A and B. The total response is decreased to 5. So instead of 3 plus 3 6, you have 5 neurons which are stimulated because of this property of occlusion. So A is stimulating 3 neurons, B is stimulating 3 neurons, and when you simultaneously stimulate, the response is less because of a common overlapping neuron here. That is a property of occlusion. So our next property is subliminal fringe. Now here the neuron A is exciting 2 neurons X and Y. Okay. Now this neuron Z, it is also innervating this neuron Z, it keeps it facilitated. It's not completely excited, but it is facilitated. So this neuron is on the fringe zone. Now neuron 6, it stimulates again these 2 neurons, but it keeps the neuron Z facilitated. So when neuron A fires, you get a response from 2 neurons, and when neuron C fires, you also get response from 2 neurons. Now when you simultaneously both these neurons are stimulated, the sum total is 5, because this neuron which was facilitated by both A and C now will also fire, because of, again, this is the property of summation occurring here. So you can see these two will, all 5 will fire when simultaneously. So the sum total of response will be much greater when you stimulate both A and B together compared to when you are stimulating them separately. So that is because of the property of subliminal fringe. This neuron Z was in the fringe zone, and now when they add it up, it will stimulate, get facilitated, and it will stimulate. So next important property is synaptic inhibition. It can be separately asked as a short note. So this is a very important property. Now synaptic inhibition can be of 2 types, direct inhibition or indirect inhibition. So first we will take direct inhibition. Now direct inhibition is also called as a post-synaptic inhibition. Okay. So this is your presynaptic neuron. This is your postsynaptic neuron. Now when the presynaptic neuron produces a neurotransmitter, and this neurotransmitter in turn causes or produces IPSP, which we have already done, that is a type of a direct inhibition, or also called as a postsynaptic inhibition. Now the IPSP produced here in this membrane causes opening of chloride channels or potassium channels, and that is the cause of inhibition. So this is a direct type of inhibition, also called as a postsynaptic inhibition, caused due to production of IPSP. Now indirect inhibition is also a presynaptic inhibition. Now here the inhibition occurs on the presynaptic membrane. Now example of this type of inhibition you usually see when here you have a exo-axonal type of synapse. So we have a nerve ending, the axon of one neuron synapsing on your presynaptic terminal, and this axon in turn, it releases the inhibitory neurotransmitter GABA. Now GABA in turn causes opening of chloride channels or potassium channels, and this in turn causes hyperpolarization of this membrane. So when the action potential arises here, there is decrease in the amplitude of the action potential because of the opening of chloride channels and the potassium channels, they decrease the amplitude of the incoming action potential, and this in turn reduces the release of neurotransmitter and hence the response in the postsynaptic membrane. So this is a type of indirect inhibition, or also called as a presynaptic inhibition. Now another type of indirect inhibition could be when your postsynaptic neuron is in the refractory phase. So whatever stimulus, it will not respond to that because it is in a hyperpolarizing state, and hyperpolarizing state it will not take the next stimulus. So that is also a type of indirect inhibition. So we have direct inhibition and the indirect inhibition. Direct is postsynaptic, indirect inhibition is usually presynaptic type of inhibition, or other cause of indirect could be when the postsynaptic neuron is refractive to the incoming stimulation. Then we have two more types of inhibition. You have a feedback inhibition and a feed forward inhibition. Now what do you mean by feedback inhibition? Now here we take an example. Here you have an alpha motor neuron. Now this alpha motor neuron, it gives a collateral to another neuron in the spinal cord, which is a special cell which is called as a Renshaw cell. Now stimulation of the Renshaw cell in turn causes inhibition of the same motor neuron which had stimulated it. So you have an alpha motor neuron which gives a collateral to the Renshaw cell, and the Renshaw cell in turn causes inhibition of the alpha motor neuron. Now the neurotransmitter release at the Renshaw cell is acetylcholine. Okay, so that is a feedback or a negative mechanism, negative feedback type of mechanism of Renshaw cell inhibition. Then coming to the feed forward inhibition. Now here we take example occurring in the cerebellum. Now in the cerebellum you have the mossy fibers. These fibers, they stimulate the granular cells in the molecular layer of the cerebellum. The stimulation of the granular cells in turn causes stimulation of the parallel fibers. Now these parallel fibers in turn cause stimulation of the Purkinje cell. Now the output of the Purkinje cell is but inhibitory. So an excitatory input via the mossy fibers finally causes the output, which is to the deep nuclei, is inhibitory. So the Purkinje cell are stimulated by the mossy fibers, and the Purkinje cells in turn cause an inhibitory signal output. So that is a example of a feed forward inhibition. So excitation, the final output is inhibition. So that is a mechanism of feed forward inhibition. So now the next property is habituation and sensitization. So first we will see what is habituation. Habituation is when we repeatedly stimulate this neuron. What will happen? There is decrease in the amount of neurotransmitter which is released from the presynaptic terminal to the postsynaptic terminal. So there will be decrease in the response, there will be decrease in the calcium influx. So there is a fatigue of the neuron. As the amount of neurotransmitter, as you go on giving stimulus, the amount of neurotransmitter which is released is decreasing, and the synapses neuron is not able to synthesize at the same speed at which the neurotransmitter is released. So that causes a phenomena of habituation, where the response of the postsynaptic neuron is reduced and finally it is completely stopped because of habituation. Then coming to the property of sensitization. Now in sensitization, what is happening? There are two stimuli. You have paired the initial stimulus with one more stimulus. It could be a noxious stimulus. So what will happen? The response in the postsynaptic neuron remains for a longer period of time. So there will be increased response for a longer period of time. Now again, this could be mediated because of increased calcium influx in the presynaptic neuron because of this pairing of the two stimuli. So that is called as sensitization. So usually the noxious stimulus paired with the incoming stimulus, the response in the postsynaptic membrane remains for a longer period of time. Now you can also have a pairing of a non-noxious stimulus. That can also cause a response in the postsynaptic membrane. For example, when the mother is sleeping through so many noises, but when the baby cries, the mother is able to sensitize the baby's voice and she gets up. So that is a pairing of the non-noxious stimulus, and that is also an example of sensitization. Okay. Now coming to the last important property is property of synaptic plasticity. So what do you mean by synaptic plasticity? It is the ability to remold the synapses. Now we have seen here normal transmission occurring from the presynaptic terminal to the postsynaptic terminal. Now what happens in plasticity? The effects are potentiated. You can see the amount of neurotransmitter released from the pre to the postsynaptic membrane is increased. Now what are the reasons, or how can you explain better the synaptic plasticity? Now people who memorize or who learn a lot revision, when you do lot of revision, what is happening? Structural changes occur in the presynaptic membrane as well as in the postsynaptic membrane, and that helps you especially in the hippocampus. Such type of learning and memory which is occurring in the hippocampus, what you can do is, as you go on revising your stuff again and again, lot amount of neurotransmitters are released, and the cause is the structural changes occurring in your presynaptic terminal. There is lot amount of calcium influx that causes release of neurotransmitter, and those effects are seen. Now if you have a short term memory, so usually in short term memory, what is happening, for example, when you remember a phone number, so those short term memory, that is because of the post-tetanic potentiation, which is a type of synaptic plasticity, but this is very for short duration, you remember certain things. So in post-tetanic potentiation, what is happening, there is increased calcium influx occurring only in the presynaptic membrane, and that increases the amount of neurotransmitter release, and these changes usually are short-lived, and that's why it is a short-term memory. Now what happens with long-term memory? When you're doing something long-term, your long-term memory, when you go on revising for, and there occurs consolidation of memory as these changes are occurring in the hippocampus, here what happens, these changes are occurring in the postsynaptic membrane. So there is calcium influx and structural changes and activation of various enzymes occurring in the postsynaptic membrane, which allows for long-term potentiation, and that helps you to remember all the things we are occurring. So your learning and memory which is occurring in the hippocampus, the mechanism involved is a long-term potentiation. So as I told you, synaptic plasticity helps you in strengthening the synaptic connections. So where we can see all these things, all the artistic people who are there, or musicians, dancers, who repeatedly practice the same thing again and again, or you are learning a new language, what are you doing exactly? You are trying to increase the strength between your synaptic connections. So that is because of the property of synaptic plasticity. So now we will enumerate the properties of synapse. So we have one-way conduction, synaptic delay, property of convergence divergence, then your property of occlusion, summation, temporal and spatial summation, habituation, sensitization, subliminal fringe, property of synaptic inhibition, which includes direct, indirect, feedback and feed-forward inhibition, and lastly the property of synaptic plasticity. I hope you have understood the properties very clearly. So please go through this again, subscribe the channel, and please share with your friends. Thank you.